Science Uncut, presented by the Volkswagen Foundation.
Arne Traulsen is a game theory expert, not a doctor or a biologist. So why invite him to a conference about the intestinal microbiome? Game theory originally aimed to develop mathematical models of conflicts and cooperations between intelligent and rational decision makers. But it has since outgrown this original scope. In what is now called evolutionary game theory, models are developed to represent populations and to understand evolution. Arne Traulsen is director of the Department of Evolutionary Theory at the Max Planck Institute for Evolutionary Biology in Plön, Germany. His talk has the title, Games Between Microbes with Rules Set by the Host. In it, he tries to bring game theory to yet another field, the interactions of microbiota in the intestine and the interaction between the human microbiome and its host. Are microbes controlled by the host, or do they control their host, is one of the questions he pursues. Traulsen gave his talk on October 9, 2014 at the Herrenhausen conference Beyond the Intestinal Microbiome, From Signatures to Therapy. The conference was organized by the Volkswagen Foundation and took place in Hanover, Germany.
First of all, thanks a lot to the organizers for inviting me. When Philip called me and asked me if I would like to speak here, I said no. I said, I have no idea. It's really very far from what I'm doing. I'm doing theoretical models, and this is a community where people know everything about the biology and know so little about the biology, it will be a bit tricky to do that. But he decided that I should do it anyway. And so the idea is that I work mostly on evolutionary game theory, and an idea that came very early on from people like John Baines was if you work on games, you work on interactions. So somehow, if you want to understand the interaction of the microbiome within the human body, somehow we should be able to use this theory in some way. And I will in my talk, I will in the first half, I will give you basically an introduction to this way of thinking and the kind of models we are working with. And then in the second part, I will try to tell you how we try to apply this to problems in the microbiome. And it's up to you if we failed or not, but I think there's still a lot to do, and there's a lot to do with theoretical models. I think we've seen some very nice examples, for example, in the talk by Eric Palmer. This is very impressive work, this Stein paper, for example, in PLOS computation in biology. We haven't gone so far because we always try to simplify a bit more. So what is game theory? Game theory is the study of mathematical models of conflict and cooperation between intelligent, rational decision makers. And this somehow suggests that I'm in the wrong conference here, because the first important book was Theory of Games and Economic Behavior, written by these two guys. And they had rational actors in mind. They thought about poker, they thought about the Cold War, in that time it was not even the Cold War, but they really thought about strategic decision-making in humans, in political contexts, and so on. And until the 1970s, this didn't really go much into biology, but then people came up with this idea we could use this in an evolutionary context. And evolutionary game theory is now the application of game theory to evolving populations in biology. This immediately means you go away from the single actor, you go to a population of actors, and you have a dynamics because you assume, I'm sorry for this picture, but the success in the game means fitness. So now you assume that people who play good, they just have more offspring, and in this way, good strategies spread in the population, and this can lead to interesting dynamics. And there are numerous classical examples, for example, hunting, group hunting in lions, for example. That's a classical example for something which you can model with a game theoretical model, but you also have games in microbes. For example, these are this is work by Paul Rainey, where they look at a quite complex life cycle, in my opinion, and they try to explain cooperation and the breakdown of cooperation in these systems. This is a very popular example of slime molds, which are unicellular organisms, and they form these weird stalks, and then if conditions are bad, they form them, and then if you're on the top here in this fruiting body, you can go somewhere else where the conditions are better. But since these are all unicellular, the ones who end up down here, they are losers. They will never have offspring. So the question is, why should you do that? Why should you use this strategy to go down here when you could be here and produce the next generation? It's a similar problem, by the way, as in social insects. So to explain why there are sterile worker castes in an evolutionary sense, why should this evolve, is an interesting question. These are all examples for interactions within a population. But of course, you could also use the same framework to describe interactions between populations. For example, here the cuckoo and its host bird. And then you can write down a game, which is a bit more complicated in this case. So this is a model to explain actually mafia behavior, where it's very interesting, I think, because these cuckoos, you could just kick out the nest, the egg from your nest. But then sometimes the cuckoo mother comes back and destroys your nest. So there's very interesting dynamics there. So why could we apply this to the microbiome even? I think because game theory is this generic way to look at interactions between all kinds of possible actors. So it doesn't have to be humans, it could also be microbes. And also it has a very intuitive part because it allows to classify these interactions in terms of cooperation, mutualism, and so on. And then evolutionary game dynamics is now a dynamical pictures where you could look at the dynamics and stability. For example, this is a system where you could ask, okay, how many types can coexist at a time, and then you could go from a kind of single type to a two-type coexistence and so on. And in this picture, we've seen similar pictures before here, actually. You could have many different stable fixed points, and you could ask, okay, there's two stable situations for the microbiome. How do you switch from one to the other? When I talk to people in your field, basically, a lot of people tell me, okay, there's somehow cooperation going on. So there's the gut microbiome as a whole does something for you. And if you say cooperation, game theorists have very specific ideas what it could mean. And so we have, if we say cooperation, we just mean something very specific. And let me use this example. So this is cooperation between two players. And let's say this guy, John, has the option to pay 2 euros for this guy to get 5 euros. And the question is, should you do that? And if I would now ask the chairman to give me 2 euros to give 5 euros to somebody else, there's basically no reason for him to do so. So especially if we make it in a symmetric way, so if we assume that these are options that are symmetric. And then if you want to analyze this, you typically write down this little structure here. You say, okay, this guy can cooperate or not cooperate, and this guy can cooperate and not cooperate. And if both cooperate, they get the benefit from cooperation, these 5 euros, minus the cost. If this guy does not cooperate, but this guy cooperates, he just gets the benefit. And this is here, he just pays the cost for cooperation, doesn't get a benefit, and here they both get 0. So the classical way to analyze the system is you think about your options. So if the other one cooperates, you have the choice between B minus C and B, so you choose B, it's larger, so you don't cooperate. If your partner does not cooperate, you have the choice between minus C and 0, so you also don't cooperate. So in all cases, you should not cooperate because that's better for you. But the problem is now, this guy is, of course, as smart as this guy, so they will both end up with 0, but where they could have gotten B minus C. And there's a long tradition of research trying to figure out situations in which this does not happen, because we see a lot of cooperation, for example, in this group hunting alliance. In general, we don't use this matrix, but we write parameters R, P, S, and T here somewhere, and then there's a certain ranking that defines cooperation. So that's a very strict definition of these things, but I think it's, at least in mathematical models, it's very important to have a strict definition on something. So otherwise you would sometimes get lost in the semantics of this. But in any case, however you do it, cooperation requires an explanation. So the argument I gave before is based on rationality. So in classical game theory, you analyze the system and you realize you would not cooperate. In evolutionary settings, you would not make this argument. So this field was basically founded by John Maynard Smith. You would say, okay, I have a cooperative system, and then there's a defective mutant, and this guy grows faster because it has a higher payoff, which translates into higher fitness, so it will spread in the population and take over. So natural selection just favors defection over cooperation. So the question is now, what are these explanations? And one explanation goes actually back to Charles Darwin, who wrote in this, The Descent of Man, there can be no doubt that a tribe, including many members who were always ready to give aid to each other and to sacrifice themselves for the common good, would be victorious over other tribes, and this would be natural selection. And of course, he writes about tribes, but when I go to John Bain's homepage, this looks very similar to me. So there are always there are tribes of microbiota sitting somewhere in hosts. And somehow, if we want to argue that they cooperate, maybe we should look at the selection on the higher level. So maybe selection is not only in the microbiome, but also on the on the whole ecosystem they form via their host. So when we have these problems, what we have to do is we have to formulate models. And one such model is given here. So you could start from a population of, kind of, say, 3 groups here of cooperators. Then you have to think about a mutation leading to a defector here, so a non-cooperator. This spreads in the population, and that's very easy. So if I have a small team and I exploit everybody, it's very easy to be successful in that team. But such a team will not be successful. So once this group is kind of homogeneous for this non-cooperative type, it's very difficult to get here. But on the other hand, if you start from defection, kind of again, the first mutation to a cooperator, this is now disadvantageous in the group. If you have a very uncooperative group and you start to be nice to people and start to cooperate, that's very difficult. But once you made it in the group, you can make it everywhere. And so the task is now to figure out what is more important. So which of these processes, how do they somehow balance each other? And you have to combine all this in terms of a mathematical model, and you have to find a measure that describes what is going on. And most of the models that we've seen here so far, they are deterministic models, which means you start somewhere, and then you know where you're going. But if you would always be deterministic here, you couldn't make this, because you can't make disadvantageous selection steps. So these models are always in terms of evolution. And if you have a smaller fitness, you can only make it in a probabilistic fashion. So what you could do is you could calculate the probability that you go from here to there, and compare it with the probability to go from here to there. And I don't want you to understand this, but this is what we do all day. So we look at these systems, we try to formulate them in mathematical terms, then we write somehow the correct solution down, and then we are very happy. But when we want to publish them, people say, this is too complicated, I can't interpret it. So we go to a simpler formulation, which is not correct anymore, but which is a good approximation. So how can we apply this kind of thinking to the microbiome? Actually, I think to me, there are some key questions that I would like to answer, and that I think that they are not completely out of reach for theoretical models. The first one is, for example, kind of arising from this multi-level idea. Can cooperation between microbes and the host be selected by higher level selection on the host level? And that's actually quite controversial, because whenever you say multi-level selection in evolutionary biology, you are kind of being attacked harshly by people, because that's a way how not to think. But maybe here it's justified. Then the question is, what is the nature of the game between a host and its microbes? Because, for example, first the microbes are interacting, and the host is interacting with them, and you could all classify this as a game, and can we somehow say something about this? So there are some early papers, actually, from 1999, is the first one by Denise Kirschner from Michigan, and she has already kind of started to think in this direction and write down Nash equilibria for these systems. And then to me, a key question is, are microbes controlled by the host, or do the microbes somehow control their host? At least mathematically, that's not clear to me, and somehow that's a question we kind of try to address. And the first one, which we could formulate here, is the host affecting its microbes? And for that, we have to write down some dynamics for the kind of microbe abundance of all types. So this is actually kind of our workhorse. This is a differential equation called replicator equation, and this is kind of I wanted to put a dot here for the time derivative, and then I thought it's easier with this one, but then I saw that you put the dot, so I should have put the dot here. Okay, so this is the change in the abundance of a certain type, and it changes proportional to its abundance and proportional to this difference, where this is the fitness of the type I, and this is the average fitness. So whenever you have a fitness which is higher than the average fitness, you will increase in abundance. If it's smaller, you will decrease in abundance. And this looks simple, but it can lead to quite complex dynamics if you want. One question, of course, here could be why don't you just use these Lotka-Volterra equations? And actually, that's more a matter of taste. So if you use the Lotka-Volterra equations, you allow the population size to change. And if you say you're not interested in that, you just normalize away the population size, and then you go from a d-dimensional system to a d-1-dimensional system, and then you end up with this equation. So if you look at the orbits of these equations, they're just identical to the Lotka-Volterra equation. But in this case, you can give some interpretation to these f's in terms of a game, which may be useful in some cases. So for this system here, we now can look at 3 cases. The first one would be you just have fixed fitness. You just have these fi's are just numbers. And then you just have growth competition. So you have different strains. They're all growing or not growing, but they're just somehow competing in growth. You could also have a situation in which these fi depend on the xi. And in the simplest way, they could just depend in a linear way on them. That's the simplest function you can imagine. And this would then be a simple game like the prisoner's dilemma I talked about earlier, so this game of cooperation. In principle, you could also assume that these fitness values depend on the time, if you want. But that makes things a bit more complicated. So we started thinking about this when kind of Thomas Bosch and people from his group came with this data, which is about the colonization of hydra. So this is this polyp hydra, and they look at the colonization of the gut after kind of starting with a sterile individual. How does the microbiome establish itself in this system? And this is the kind of data they gave to us, which is kind of, they have a number of replicates here. Here's 3 and 4. They have a number of weeks, and every week they do a measurement, and then they have lots of kind of different types that we should look at. And it's for us, it's very hard to make sense out of this kind of data, because what we prefer is something different. Because here, if you have many types, if you have 2 types above here, you have 4 parameters. I needed these 4 parameters for my payoff matrix. If you have n types, you need n times n parameters. So if we have these 200 bacterial types in the average human, we have 200 times 200 parameters, which is a real nightmare. And so we don't want to have too many types. So the data that we would prefer is actually quite different. So the data we would prefer is kind of fewer bacteria, but many more time points. We want these time series that many people talked about here. Because if you really want to make a predictive model, you have to understand the dynamics of the system. And it's not enough to look at these snapshots. So the conclusion from the data at that time was, okay, there are many out there, and we can assign names to them, and we may even know what some of them are doing. But it's not really what we need to make a mathematical model about it. And from a theoretical perspective, we would like to have many more time points, many independent replicates. So you should start the system in the same condition and then just look at its evolution, for example, to figure out how stochastic it is in the end. And also we would like to have few bacterial strategies because we can't deal with so many. So this is an example from another field where I'm playing around. This is cancer modeling, and of course, this is what we want, right? We have a time series here. It's only 5 or 6 time points here, but we have a mathematical model which fits this in a mechanistic way, and then we could make individual predictions. And this is the ultimate goal in this field to me as well, to be able to make a prediction for an individual. But we're still very far from that. So we looked at the data again and again, and then we decided, okay, if we want to do anything, we have to focus on a small aspect of the data. We cannot take all the data together because we also don't know how reliable it is. These low abundance types, are they really is this really true, or what's the measurement error here? Do we really have to model something that has an abundance lower than 5%, and why? So if you look at this long enough, you always see this pinkish type here, which is always it always grows first, then it goes away, and then it grows back. Goes in, out, and in again. And we call this in-out in dynamics, and you can also see that if you look at the similarity to the adult. So if you look at the age here, so the individuals become very close to the adult after about 2 weeks. Then they go away again, and in the end, they go back to this adult thing. Not knowing anything about the underlying biology of this, it's still interesting to figure out what is the mathematics of this. How can I write down a mathematical model that shows this kind of dynamics? And now I could go to this replicator equation, this differential equation that I showed, and I could just ask, okay, what do I have to choose? What kind of fitness do I have to choose to explain this? And the first case would be fixed fitness. This is this growth competition. This is certainly not enough. It's easy to show that this cannot explain it. Then you could ask, okay, if I have only 4 types, and I assume a simple game, then I need only 16 parameters, which is quite a lot. But can I somehow show that with these 16 parameters I can make it or not? And it turns out this cannot explain the dynamics. So you need even more than these parameters. So if you have n types, you need more than n squared parameters for some reason. And you could also say, okay, let's take the data and take it serious, and let's assume that we just have a time-dependent fitness. So we vary the fitness with the age of the organism. This is good, but it can explain everything and nothing. So then the data is better than the model because you put everything in there already. So it doesn't really help. And that's actually a trap that we often fall in when we try to set up models and work close together with biologists. Because the map is not the territory, which goes back to this guy. And the idea is you want to build a model to explain something, but you don't want to have a realistic picture of reality. So that's actually a very nice fable by a guy called Borges. He's Spanish. I can't pronounce the name, I guess. Which is it's about cartography. And it's called On Exactitude in Science. And the idea is kind of, okay, the story is there's the cartographers, and they are building better and better map, and in the end, the map is as big as the kingdom. And then it's completely useless, because what should you do with it? So the map is not the territory, and what you want to have a simple model that explains some things here. So is there something in between? So not in this time-dependent fitness values, but not in the but that could still explain the data somehow. And yes, you could assume that this is your replicator equation, and this is something which is normally called a mutation term. So this is now the replicator-mutator equation. But there's some parameter here which determines how often types that were absent before can come into the system. And if you assume such a term, you assume a simple function of this, like an exponentially decaying function, for example, then this can capture this in-out-in dynamics. So this is one example where you go in, out, and in again. So you need some time-dependent factor here. And our kind of idea was, if there's a time-dependent factor here that is external to the system, so external to the microbial interaction, this could somehow, in the long run, be the basis to show that the host must do something with it. But there are many questions to be answered yet. So an empirical question, for example, is the dynamics of colonization time or age dependent? So if we take this microbial system at another time, do they still interact in the same way, or is there something that is more complicated? And if this depends on the whole history of everything, then this becomes very, very difficult, because you need very different models. And basically, you can explain everything and nothing then. And there's an interesting theoretical question for the mathematicians in the room. So can we mathematically prove that a time-dependent factor is necessary to explain this in-out-in dynamics? Because I'm a physicist by training, so to me it's obvious that it's true, but a mathematician will say, wait, you have to prove this, and this could take a PhD thesis or two. It could take a long time. It's not an easy question, but still it would be nice to know this. Yeah, apparently nobody's jumping and happy that you could do it now. Okay, and then there's an empirical and theoretical question. Are these pairwise linear games even a good model to think about interactions between microbes? Because we have no idea. I mean, the Lotka-Volterra system is very close, but is this a good model to start thinking about the problem? And so this led us to a follow-up project with the group of Thomas Bosch, where we'd ask, can we actually understand these bacterial interactions somehow, and how do they interact? So how do they interact? So people typically show me these pictures, and of course, whenever there's an arrow, I want to have numbers. So what we want to do is then we want to write down these systems, but the conclusion is somehow it's complicated. If you really want to understand all this in all its dynamics, I think it's easy. It's relatively easy for you to give me the parameters. So all these numbers here for these reaction rates and so on, this is something in principle you can measure. But what is more difficult is these functional forms, because in all these models there are assumptions on functional forms. So how do things change with something else? And that's much harder to figure out. So this conventional model would just require a huge number of parameters and detailed knowledge. So what we thought we could do is we could just ask the simplest possible question, is if you just have interaction between two types. You just take two of these players from the hydra gut, and then you let them grow together in isolation. And you ask, do they somehow affect each other or not? And the choice of bacteria was obviously not done by us, but by the experimentalists. And they said, okay, somehow you should take important players, so things that are really there and not obscure things. And something that is, of course, somehow distinguishable by colony phenotypes, because then you can plate them and look at it. And there are, of course, many issues with this approach. So the interaction will depend on the medium, of course. And then the growth dynamics will not reflect the dynamics within the host, because there are, of course, more different microbes, and the host is somehow different. It's maybe closer to a chemostat than this system. So there are many issues, but still, let's try and let's see if we can learn something from that. So this is kind of the time here in hours, and this is the frequency of one of these species. And you realize if you start with a high frequency, you stay there. And if you start from a low frequency, you go down. And if you look at this problem from this level, it just looks like a bistability. So you go up when you're above a point, and otherwise you go down. This is a classical economic problem. It's called a coordination problem. And it's the same problem that you have if you drive on the left or on the right-hand side. So if you move to England and you continue to drive on the right-hand side, you don't have much offspring. And that's the evolutionary picture is just... can we understand this in more detail? Because this is certainly not sufficient. And is the growth of the bacterial populations of any importance? Because all these models that we have, they consider constant populations, or they consider very large populations and only the relative abundances. And we have to go beyond these relative abundances to understand more about this. So the first question is this really frequency dependence, which is what we are looking for, so dependent on the relative abundance of types, or is this just density dependent? So what our collaborators did, they looked at the kind of different densities and at the growth rates, and they found that this D type is always a higher growth rate than the C type here. So there is no density dependence on the initial condition here. Then you could look at the frequency of one of the types and at the growth rate here, and then you see that one of them is relatively flat. All these green symbols are on this line, but the blue one shows this nice maximum here, which could be fitted, for example, like in this case, by a parabola. So somehow their interaction now depends on the types, on the frequencies. And in these games that I talked about, so I wrote down this big matrix or this little matrix. This is all kind of leads to these linear interactions. So this would mean that the blue curve should be something that is a line that crosses somewhere here, then it's interesting. But it's a parabola, which means if you take it serious, if you take this as fitness, it means you have a quadratic frequency dependence, and then you have something which is typically called a 3 or 4 or 5 player game. And I've actually cheated. I've shown you this. So there are many stable fixed points, and this only arises if you have more than 2 players. And you can show that kind of how the dynamics becomes more complicated. You can do all the maths for this, but maybe it's not necessary. Maybe you could just go back to your Lotka-Volterra equations here. Just say, okay, there are 2 parameters here. So this is the change in abundance of type C. This is the growth rate, and this is the carrying capacity. And the carrying capacities are shown here, and they are relatively stable. So the point is kind of these growth rates, they depend on the frequencies. If you just plug in the functions that we found on the slide before into these equations, you find these orange arrows here. So you always go to this red line here. And if you compare the experiments now, which are the blue and green symbols, so you start somewhere down here, and then in some way you go close to this point, which is a stable fixed point, or you go kind of close to this point, and then we don't really know what happens because the experiment ends, but we should actually move somewhere else in the end. So the point is here, you can now, based on this model where you took just the growth rates as a proxy from the system, you could try to understand the dynamics of this. But this is only for 2 types, and this is already a lot of work. And I think if we want to go to many types, it's much more work. So we have to do something else. So we could, for example, take an approach like the Xavier model, where they basically kind of try to fit everything at once. But it's also a bit tricky because if you want to fit many, many parameters from a time series, it's not so easy to figure out if this is the only solution, if these are the right numbers, and so on. Okay, to summarize, so evolutionary game theory can be studied to study interaction within and between evolving populations of microbes, birds, anything. Then this colonization dynamics can only be described by this time-dependent factor. So we need some time dependence in our equations. And to me, still an open question is how can we really understand the interactions between growing microbial populations? And one thing that I didn't really mention is that all these concepts like cooperation, mutualism, and so on, they're typically defined in a constant population. And it's very hard to define something like cooperation in a growing population because somebody who is growing fast but is at the same time increasing the carrying capacity of the population in a very fast way, would this be a cooperator or a defector? That's not so clear. Okay, I should thank people from my group and also kind of experimental collaborators from Kiel for allowing us to work on this. And yeah, thanks for your patience with me.